Yeah, so let us begin with the class for today. Then I'll be sharing my screen. So just let me know if everything is working well. Is it okay? So open the slides. Yeah, okay, thank you. Yeah, so again, this is the final TA class for me that will be for the course Bioinformatics Algorithms and Applications instructed by Professor Michael Gromiha. So uh, within the 12 weeks of the lecture series by Professor Gomiha, so he has covered a lot of concepts that range from, just core understanding of bioinformatics to, delving into different fields specifically that comes within the broad kind of umbrella of bioinformatics. So it's whether that be from database to web servers to different sequence analysis, phylogenetics, and structure-based analysis, drug discovery, etc. So all of those are covered within this 12 week lecture series videos. And in the last kind of TA class for me, so what we started was that we started recapping, right? So we started recapping what we have gone through, like briefly over the past TA sessions that occurred. And then we are in the middle of that. So we, I think, covered until the Needleman-Wunsch algorithm for global alignment, right? And then we said that we will talk about the rest of the concepts in this class. So this will be the final additional class kind of for this course. Yeah, so let us just start with a brief recap. So I'll just go through the slides quickly. So this is basically the fundamentals of bioinformatics as was explained earlier. And then the various structure, sequence, and functional aspects of bioinformatics analysis. The significance of the studies, the five kind of main pillar of bioinformatics, and then we looked at different structure-based parameters, sorry, the sequence-based parameters in the case of your protein sequences, amino acid sequences, as well as in the case of nucleic acid sequences, right? So in the case of amino acid sequences, you have the analysis of something like charged amino acid content, maybe hydrophobicity, these kinds of parameters you can do. You can calculate, and then for the case of the nucleic acid sequence based analysis, so what we are going through are the different again parameters based on the dinucleotide or trinucleotide sequence, right? So those parameters have been calculated for a kind of short range of subsequence, and then you can use a overlapping sort of window method. So overlapping sort of window of 2 or 3 nucleotides, and then you can calculate the overall average value for these any parameters. For example, it could be rigidity, it could be flexibility, the melting point, again stacking energy, etc. So these are possible for the different nucleotide sequences. Yeah, so we talked a little bit about how NGS is important due to its large data sets, and this is what basically I was talking about. The different dinucleotides and trinucleotide sequences that are possible, and the parameters that you can calculate based off of that, basically, right? I think there was one short chart where you, yeah, this one. So these are kind of the examples of what properties you can calculate if you have the values for the individual dinucleotide or trinucleotide parameters, right? Sequences. If you have value for all of the parameters, you can calculate it. And then we talked about increasing the number of nucleotide steps that you can take in a calculation. So that's not always the better idea, because even though you get a much larger number of possible nucleotide steps, for example, if you go for tetra or pentanucleotide, it's almost always the thing that uh, a lot of those pentanucleotides or tetranucleotides that are possible, you will not have any parameters measured or calculated for those. So in that case, due to the unavailability of large amount of data, your calculations will kind of be void in that sense, right? So for example, in this case, you see that already some trinucleotides, they have only a limited number of samples for certain parameter calculations. So increasing the number is not always good. You can keep it between 2 and 3 so that you can get a good accuracy, as well as that you have enough number of data, enough number of samples basically to have your data, right? Yeah. So these were the things that we went over: sequence-based parameters and the numerics calculations. Then we briefly went over databases, right? The biological databases, for example, primary and secondary databases, and then briefly over the structured query language, what it indicates, and then mainly where it is basically integrated. It's integrated in the relational database. Relational database have basically the databases where you have your data organized in the form of tables, multiple tables, and then two tables, for example, they will have one common columns. So this way you do not need to parse through all of the flat file in order to get whatever um, kind of data that you are getting, right? In order to get whatever information or query that you want. But it will basically make it so that only certain tables you have to go through thoroughly, and then based on the output that you get from the table, so you can directly search for those in other tables, right? So there is another concept which is called object oriented database. This is kind of a little let's say latest version that can be derived from relational database, kind of using those concepts where every uh, information, you can say every information in the database is kept inside something called objects, and different objects, they have pointers connected to them, so that you can trace information from one object to another. So that makes executing your searches or queries easier, basically. And then we talked about weekly drug discovery, and then yeah, finally this is where we were earlier in the part of sequence alignment, right? Sequence alignment is quite crucial, because it can not only give you idea about the overall alignment, which is important in a lot of cases. If you want to see similarity, if you want to see evolutionary relationships in phylogenetics, those are important, but you can also do local alignment. So local alignments are important in the cases, for example, in motif discovery, where you have a small motif in a certain part of the sequence, and based on um, the different sequences that or the different motifs that we get from sequence alignment, so you can discover basically functional regions of different proteins, right? Or in any kind of sequence. That was it. We saw that the brief or the most simple visualization technique for sequence alignment, that could be a dot matrix, where you match the characters of a sequence with another, and then where you find a match, you just put dots, right? You can do it just raw, one character by one character. You can put a window or stringency, window size along with a stringency, to basically minimize the number of dots or reduce the number of dots that you have, which can sometimes give you better clarity in your plot, in understanding your plot, basically. Okay. We saw examples like this one, where you have a lot messier and cluttered plot with a lot of dots on the left hand side, whereas on the right hand side you have basically kind of a clear plot with certain basically what do you say, certain patterns clearly visible here. For example, this is sort of indicating repeats, right? Repeats within the sequence, within this particular region of the sequence. So it's quite handy in a lot of cases. And then we saw that while dot plot can give you a good visualization for your sequences and their alignments, to particularly quantify what will be your optimal alignment, the most optimal one, and then what will be the net alignment score, you need to, of course, introduce some sort of scoring, right? And then we saw that if you have a match between characters, you can introduce a match score. If you have a mismatch within the characters, you can introduce a mismatch score. And if you have a gap, you can introduce a gap penalty. So the gap uh, is basically not favorable by evolution, more so in the case of a origination kind of penalty, which is the origin of a new gap. So in those cases, basically the sequence alignment is penalized in certain scoring systems, in the scoring kind of methods, where if you have basically new gaps introduced in the alignment, then those will be receiving negative scores, basically, right? And then based on that, you can identify what is the most optimal alignment. But again, these are just examples of simple scoring systems. It doesn't always happen that a match score is given as 1 and a mismatch score is given as 0. Instead, while trying to align or compare different sequences, what you can do is you can retrieve these scores from some matrices. And we saw that two of the widely used matrices are PAM matrices and Blossom matrices. So PAM matrices are basically uh, constructed using a homologous set of sequences with greater than equals to 85% similarity. And what we basically kind of construct first is a PAM1 substitution table. So PAM1 unit is defined as 1% of amino acids that have undergone changes. And so PAM1 you first calculate, and then you do matrix extrapolation, basically the score extrapolation for the higher order PAM matrices by multiplication. So you multiply PAM1 times PAM1, you get PAM2 values basically, right? And the further you go, you will see that the mutation rates, they will kind of increase sort of the scores that you will get ultimately upon converting the scores to your logarithmic forms and doing the normalization and stuff. You will see that ultimately the scores will increase. So if you go from PAM1 to higher, so your mutation rate is increasing from one amino acid to another. So that means that PAM1 is better suited for a closely, of course, this is very closely related homologous sequences, but any lower kind of PAM values, let's ideally around PAM 40 maybe. So those are suited for basically closely related set of sequences. And then if you go for even higher, for example, PAM 200, something like that, so that will be suited for distantly related sequences. So you can get the match and mismatch score from this kind of matrices, PAM matrices. And since PAM matrices were basically the higher order values, they were extrapolated, not directly measured or calculated. So to kind of combat these, the Blossom matrix that was developed. So these are directly basically calculated from observations. Okay, so observations, that means um, it can kind of correlate with the real data basically. And the order of how you use the PAM, the Blossom matrix differs. It's basically the reverse of what you do in a PAM matrix. So PAM lower score, that will be for closely related sequences, whereas the Blossom lower scores, that will be for distantly related sequences, basically diverging sequences kind of, right? So that was the Blossom matrix. And if you can look at the scoring table, they kind of look the same, very similar in case of PAM and Blossom. So you will see the positive scores. They will indicate the kind of, you can say, favorable mutation, as well as for the same versus same amino acids, you will get a positive score because that will indicate the same amino acid staying the same, that propensity. And then the negative score, they will indicate kind of unfavorable mutation, if you will, right? So based on that, of course, you can get your the simple scoring methods basically. But how you kind of alignment do the alignment from the perspective of a global alignment or local alignment, that comes down to two different widely utilized algorithms, you can say. So one was Needleman-Wunsch algorithm, which we discussed just in the earlier section or in the earlier class. So basically you first initialize the algorithm with a 0 on the left hand side, top left cell, and then you go horizontally and vertically and fill out the first row and the column with accumulating gap penalties. Basically, if the gap penalty is -2, then you will just do -2, -4, -6, 8, -10, etc., etc., right? And then you find out the score of the next cell by calculating the score from these three different paths. One would be the diagonal, another would be the upside down arrow, another would be the left to right arrow, right? And then based on the best scoring, you will get the score for that particular cell. If you fill out the entire cell, the top right value for the cell, that will be the net basically alignment score for the Needleman-Wunsch algorithm. And while doing the sort of the scoring steps for each cell, you have to give it an arrow to finally be able to um, trace the path that a certain cell has received a score from, right? So in this case, if you trace the arrows, so you will get the optimal alignment. Wherever you have the diagonals, so that means that those sections will be aligning. Wherever you have a vertical or horizontal jump, that means there will be a gap. And you can figure out easily where the gap is, because you will see that the characters for one sequence is moving forward, whereas for the other sequence that is not moving forward. So in that case, there will be gap and you can place it accordingly. And that was the Needleman-Wunsch algorithm basically. Now, for the local alignment, of course, we have the Smith-Waterman alignment matrix. So this is quite the same. However, instead of putting a negative value wherever you encounter or wherever your calculation gets you one, you just put a 0 instead of that negative value particularly, right? So for example, your initialization will also work that way. And then you can calculate based on the same three different routes, the arrows and the horizontal and the vertical jumps. And then if you go on calculating, then ultimately we'll reach the kind of blocks that you want to calculate the value for. And if you fill out the matrix basically, so for example, in this case, if you fill out the entire matrix, you will see uh, that for certain cells, of course, you will get positive values, right? So initialization was done with 0. Then through the different routes, you calculate all of these values. And the entirety of the matrix when filled out, you will see the certain cells have positive values, right? So for example, the bottom right one, it has a value of 1. So do you remember what would be the net alignment score for this Smith-Waterman algorithm kind of matrix? So it looks.
Sir, 4, I guess, as a base because in the last
So in this case, not yeah, in this case, not the right bottom score for the cell, but in this case, what is the maximum value that you are getting within this matrix? So that will be your optimal alignment score, net alignment score for the Smith-Waterman. And again, as usual, you just do the tracing of the path that you got the scores from, and then you will ultimately reach the alignment. For example, this is 4, there is 3, there is 2, there is 1. So I guess this is the kind of the diagonal where the alignment is occurring. And then you will see that corresponding to this, you have C, G, T, G, A, right? If you go, yeah, GTGA. So right until not do not like include the 0, but there is 1, 2, 3, 4 here, right? GTGA. If you go on the left hand side also, you will see GTGA. So that means that these two regions are kind of matching. So that is your local alignment. So you have found a particular region which are aligning between these two sequences. So that might be some sort of sequence of importance, so GTGA, right? Just yeah. Yeah, so that was one type of analysis, your local alignment algorithms. Now, what you can do is you are aligning certain parts. You are finding out the whether the whole sequence alignment or whether certain parts or subsequences are kind of matching together, right? But there is another sort of particular analysis where you, instead of just doing sequence, the counting on the entire thing, you basically do a positional analysis where at a particular position in the sequence whether certain amino acids can be conserved or not. Okay, so that is what this conservation score is talking about. So it's talking about unweighted and entropy based measure for position 6. So in this particular position 6, whether the amino acids are conserved or not. So that is what we wanted to say. And basically the formula goes kind of like this. So it's Fa of I times ln of Fa of I. So in this case, what your conservation score will be, so what this I will be, it will be fraction for all of the amino acids. So all of the amino acid categories that you have. For example, here we have a leucine, we have an alanine, we have a valine, etc. And then basically how many number of times you see in all of your sequences, right? So out of the 5 sequences given, we only see each one time. So in this case, the fraction will be 1 by 5. And if you do the kind of calculation, it's 5 times 1 by 5 times ln 1 by 5. So it will give you a score of minus 1.609, right? Yeah. So these kinds of formula you should try to remember. Again, for example, in this one, in position 9, you have the same thing basically. So remember that this fraction is basically out of all of the sequences, how many times you see a particular amino acid at this position, right? So for example, in this position, threonine is basically seeing 5 times, right, out of 8. So that will be 5 divided by 8 and 5 by 8, and the rest of them are kind of the same. So it will give you minus 1.073. So in this kind of way, you can calculate the conservation score. We looked at briefly what kind of amphipathic means. So that's basically alternating kind of hydrophobicity, hydrophilicity. You can see it in a lot of cases. For example, a lot of proteins, they are kind of necessary for their function to have this sort of structures or structural orientations. For example, in heat shock protein or in this PL IN4, some interesting applications are there. Sometimes if your kind of lipids are missing from the bilayer, so it can introduce this sort of amphipathic region of your protein inside the kind of lipid bilayer, as you can see, lipid layer basically. So that will maintain the structural integrity. So there are some instances where it's quite useful to have an amphipathic set of um, basically amino acids in your protein structure, basically, right? Um, yeah. So again, so we talked about the alignment matrix. Now this times there is a weight matrix basically. So weight matrix is kind of the same. You have to first get the alignment matrix by counting uh, basically at each of the positions particular amino acid and the count of that particular amino acid. For example, in position 1, alanine is given 3 times. And then you do the counting for all of these. You fill up the matrix for alignment, and then you just identify or you just add this formula to get the weight matrix. So it's kind of the same. Just revise the old slides that we have uploaded here. And then based on that, just try to remember some of these formulas, because these are the formulas that Professor Tomiha talked about. So these are sort of important in that sense. Okay. And another similar concepts were position specific scoring matrix. Um, Again, it has a positional information of an alignment. And what basically happens is that uh, you have to firstly convert the multiple sequence alignment that is given, for example, here, to a raw frequency table. So the raw frequency table will be at position particular, for example, position 1. What is the frequency of getting an adenine, for example, right? Adenine, thymine, guanine, cytosine, and so on. Then you also have an overall frequency. So this overall is basically considering all of the positions. So for example, 5 times 6, 30, right? So out of total, out of that, basically, how many times you are getting this particular, with this particular nucleotide. And then you have to do the normalization, where you uh, basically divide each value for individual frequency calculation, for positional frequency calculations, by you have to divide those by the overall frequency for that sort of nucleotide, convert that to log base 2, and then that's how you get your position specific scoring matrix for all of the kind of positions there, right? So you can try to remember this as well. um, and then since on the amino acid side, we talked about certain calculations that you can do. So those are basically hydrophobicity calculation was one of them, right? So hydrophobicity indices of the residue, if you plot that against the sequence number, so that will give you something called a hydrophobicity profile, which is basically the same as uh, getting the idea for all of the individual residues together. So you will plot all of the residues and their hydrophobicities together. So residue number on the x-axis and then hydrophobicity on the y-axis. And based on the structure of the plot, you can say whether the residues have repeated pattern of hydrophobicity and hydrophilicity, or whether a certain stretch is all hydrophobic amino acids. This kind of basically information you can get from a hydrophobicity profile. So anytime the profile part is mentioned, it is usually against something like residues, and since it's hydrophobicity, so it will be the hydrophobicity profile, right, based on the indices, right? And we also looked into how you can normalize certain values and keep them within basically a small range. So that is a lot of times helpful in your while you are trying to basically train different models. That can also be helpful, because by shrinking the range, you can improve the accuracy of the model sometimes, right? So here, for example, the normalization formula for this set of values, it was x minus mean divided by max minus mean. So this is the one for particularly normalizing between 0 and 1. As you see, if you do the calculation, so 2 in this case will become 0, and then 20 will become 1. So remember this formula uh, as it was seen in earlier assignments also. And then we saw uh, basically for any kind of plot, be it your dot plot, be it your hydrophobicity profile, or any kind of profiles. So if you have kind of the max, now if you keep increasing the number of window size, so window size will mean that instead of doing something, instead of plotting something character by character, you do basically consider a collection of residues in this case as a single sort of point, right? And you do their hydrophobicity calculation, average hydrophobicity maybe, and then you put a dot on the plot basically there, right? So in this case, as always, if you increase the number of window size, or if you increase the number of residues that you take as a single window, so that will basically smoothen your plot more and more, basically, right? So in this case, 15, which is the highest uh, number given as the option, so the smoothest plot can be obtained from a window size of 15, basically. So the higher the window size is, the smoothest plot you can get, the smoother the plot will be. And this was just a capping of the dot plot, as we have seen the same behavior there also. Okay. And then we talked about the UPGMA method for constructing the phylogenetic tree. Now, the phylogenetic tree was basically a tree that describes evolutionary relationships, right? And in this case, if you have multiple taxa or multiple species, kind of, let's say you have their sequences, right? So, firstly, what you do sort of a distance calculation, a combined distance calculation between the different possible pairs um, in your analysis, let's say in your data set, and then based on this particular small method, you do the UPGMA based phylogenetic tree calculation. So firstly you do the initial pairing, then you have to place the node at the midpoint between the two taxa. Which two taxa? Basically where you get the smallest pairwise distance, right? And then now what you do is that you consider these two taxa as one composite taxon, single taxon, and then you do the distance calculations again and again, right? And if you keep doing that, you will ultimately reduce uh, the number of distances that you have to calculate, but also at each kind of step at this UPGMA, you can find out which two taxons will go together basically in a distance based way. So just for the examples here, we had A, B, C, and D. These were the four species. And let's say you have calculated their distances. You have done some normalization, log-based change. And now you have the pairwise distance as these, right? 0.40, 0.35, 0.7, and so on. Now, see, it seems that out of all of these, 0.35 is the lowest value in this set of values, right? Which is between A and C. So what we will do is that we will place a node at the midpoint of A and C, and we will just consider, okay, A, C. Now I consider them together as a single composite taxon, and then the distance from the node to the midpoint, that you can also calculate just by dividing it via 2. So 0.35 by 2 is 0.175. So that's done. And now you do the calculation of this matrix again, where you have AC as a single composite taxon. Now you need to calculate the distance between AC and B, as well as AC versus D, right? How you do that? You just do an average of AB plus CB, uh, basically AB plus CB divided by 2. So that will give you the AC versus B distance. If you do AD plus CD divided by 2, that will give you the AC versus D distance. And then the B versus D distance stays as it is. And now from this new set of matrix, you see that, okay, so the AC composite taxon uh, is AC composite taxon that is in the closest proximity of B, as you have the lowest value of distance there. So now you say, okay, so AC, we had a single composite taxon where we assume that A and C are kind of staying together. They have the lowest distance. Now that single taxon is connected to B, and you place kind of a node at the midpoint here. You have the distance as calculated here. And now you take BAC as a single taxon basically. Now the BAC versus D distance, again, you can calculate just by doing the averages. And that will give you the entire phylogenetic tree, where first of all you have AC, the closely related species. Then with the composite AC, you have B as the closely related one, and then with the composite ACP, you have D as the closely related one. So that's basically the short way kind of constructing the UPGMA based matrix and the UPGMA based tree for phylogenetic tree calculation. And how you can represent a particular phylogenetic tree, you can do the Newick formation or the Newick notation for representing any phylogenetic tree. Basically, whenever you have two species that are connected via a node, you can put a bracket uh, and you can keep the two species within that bracket. So that will give you sort of single composite, if you say, right? For example, here AB you can keep within a single bracket. Then you also have DE, which you keep within a single bracket. And interestingly, the composite of AB, the composite of D, and the composite of C, they are also uh, kind of kept within a single bracket, basically, right? So you have A, B, C, D as well as A, because they are all connecting together here, right? So in this way, just by putting brackets between the taxon and the composites that you that are connected by nodes, you can convert the Newick notation to a basically phylogenetic tree or vice versa. Yeah, so you can calculate the average hydrophobicity also, just a simple numeric calculation, uh, plus all of these, and divide that by the number of total amino acids here. And yeah, one another way of constructing or just doing the notation of phylogenetic tree was, instead of assuming equal distances between, for example, a shared node and then two different taxon. So if you have mentioned the distance or distance ratios or something like that, so you can put that into the phylogenetic tree with a colon, basically, right? So for example, B colon 1 and then C colon 2. So these will be within a bracket. So that means that from this particular node or the branching point, C is twice as distant as compared to B, basically. So that is what it means, basically. And then we saw that depending on how many species you have, you can either construct rooted trees or unrooted trees. So the rooted trees, they will have a defined origin point, kind of, whereas the unrooted trees, they don't have a defined origin point. And just remember these ones, because that is also critical formula. So the number of rooted trees for n taxa that you can use, that you can calculate using this formula, and the number of unrooted trees for n taxa that you can also calculate using this formula. Okay, so just quickly remind me, which number will be greater? Are these the rooted tree number or are these the unrooted tree number? So if you have a specified number of species, then are the number of rooted trees that can be constructed greater, or are the number of unrooted trees that can be constructed greater? So it's basically,
Sir, rooted trees?
yes, rooted trees, they are kind of greater, right? So if you see the examples, for example, if you have three taxa, so rooted trees can have three topologies here, whereas unrooted tree, you only have one topology because the origin point kind of isn't there. In the case of four taxa, you have the rooted trees, unrooted trees, you have three number of topologies. For the rooted trees, you have 15 number of topologies. So always the rooted trees that is higher. Basically, if you consider this number of taxon versus the number of topology of the trees, you will see the rooted tree is always a little bit higher than the unrooted trees. And as well as you can see that if you keep increasing the number of taxon, then the number of the tree topologies, they go up exponentially, right? It's needing more computational power, basically. We talked about bootstrapping is method for evaluating confidence level. Um, and then we saw again amphipathicity. Amphipathicity is basically a periodic nature of polar and non-polar residues. And yeah, these kind of simple calculations you should remember because you can get it quite easily. The composition calculation, the hydrophobicity calculation, so on and so forth, that you can just simply do from a sequence directly, right? As well as your the GC content calculation, etc., from a nucleotide sequences. Okay. Yeah, and then we saw that in certain cases you can get a little bit more analytical type of questions where you have to kind of compare and contrast the correlation or something like that, any parameter like that, between different groups to identify or reach a conclusion, basically. So here again the same thing. It asked in terms of the deviations, right? So the deviation where that is larger, so that will mean that it's more distant, basically, where the deviation value or the parameter is smaller, so that means that it is closely related. So in this case, again, based on the composition, option B or group B, that is closely related with the unknown protein, basically. This type of analytical questions you can also get. And then, yeah, we briefly talked about what non-redundancy is. So non-redundancy is basically getting rid of all very similar data from a data set, which sometimes can bias your calculation. So for example, just this was given by Professor Promiha himself. So if you are calculating composition from a set of certain number of proteins, let's say certain number of sequences, you do composition calculation for alanine, you find 0.20. Now you add one particular sequence which is very similar to one of the earlier ones, and then you see that the value is skewed kind of here, right? 0.23. So in this case, since you have a more alanine dominant sequences that is introduced, which is very similar to one of our earlier sequences, so the composition of alanine goes up. While on the reverse case, the composition of alanine will go down if you do introduce a sequence which is less dominant in alanine, but is also quite similar to one of our earlier ones that we already had. So in this case, you can introduce some sort of cutoff that, okay, if you have a particular sequence, so you do some calculation of composition, let's say you put a difference calculation, and then let's say based on a certain level of differences, you decide whether you want to put it as a new cluster or as a new point in your data set, or whether you want to just say that it belongs to kind of this group of um, amino acids, right? And based on that, you can select a representative from that group that will keep your data set less biased. Basically, we saw one particular example software which is called CD Hit. CD Hit basically again tries to do the clustering of amino acids, um, and it does it so via something called the word filter or the short word filter. Basically, if you have uh, amino acids, you can think of them as characters, right? So if let's say region of sequence of amino acids is matching together, so that means that there must be some short stretch of characters or words. They are also matching between these two characters. So that is how they do it. There is a threshold and a limitation for the short word filter, because it can happen that sometimes, so let's say just this example, protein A versus protein B. So if you have 50% matching between protein A and protein B, and if you do a two-step word filter, so basically two-word filter, so in the worst case scenario, what can happen is that your matches and mismatches are uniformly distributed. So in this case, uh, the word filter cannot work because you cannot find a two-stretch character where it is matching together between the sequences, right? So in the case of uniform distribution, it does not work. So that is kind of the worst case. And based on that, certain thresholds have been devised for all of the word filters that you can use in CDA, right? So that was one thing. Just again, since this was also a tool that Professor Tumia explicitly kind of discussed, so just remember these terms. Okay, so in any case, you have a input file which is always indicated using -i in Linux, right? If you have an output file that is usually indicated using -o, and then in this particular case, you have the -c and -n, which are specific to this particular CD hit tool. So -c of 0.9, that is related to the software, that indicates 90% identity or the clustering threshold, and then -n of 5 that relates to the word filter size or the word size, right? The rest of the kind of um, the rest of the parameters that you are indicating, those you don't need to memorize, because sometimes those are memory or CPU related, basically, right? So -c and -n, you just remember these for the for the CD hit tool. And since we were on the top of clustering, because CD hit does combine different amino acid sequences via clustering, right? Using its short word filter. So you also discussed the k-means clustering. I think this was also discussed briefly by Professor Promiha. So what k-means clustering does is that uh, it tries to group a set of data into a specified number of clusters by, let's say, measuring their distance in one way, right? And then it does the iterations where it basically does to compute the best available options um, by constantly reassigning in each of the new steps what should be the new centroid of the cluster and how should the distance be from the new centroid to the available data points, and then based on that it kind of regenerates the area that a particular cluster should cover. Okay, so for simplicity's sake, we did kind of a 1D example, 1D clustering example. So we said that by examining or just simple visualization, you can see three sort of clusters are there or possible, right? But computer, of course, cannot just see it directly. It has to do some calculations. And how the calculation is done in the k-means clustering method, it particularly, firstly, you have to define the k value, which is the number of clusters uh, that you want to divide your data points into. So if you give it a command to select 3 data points, what it will do is that it will randomly, out of all of the available data points for clustering, it will randomly assign 3 to be the part or the centroid of 3 different clusters. And now if you have the cluster centroid, which are 3 random data points, and now you try to assign all of the available or all of the remaining data points into one of these particular groups, whether blue, green, or red, right? How you do that? You measure the distances. Whichever distance is less for a particular data point, you just assign this data point to be a part of that cluster. So in this case, the blue distance is smaller. So that means this data point is also part of the blue cluster, right? You do the assignment for all of these. Now you have some sort of assignment here. And then how you proceed for the next step is that you do find a new uh, point to be called the centroid of the cluster. So how you find that? So this is the uh, basically the average kind of position of these 2 data points. So that will be the centroid or the new centroid of the data for the blue cluster, right? Similarly, we have for the green, for the red also. And now you do you iterate the process again by reassigning each data point to be part of the particular colored cluster. And at some point, you will see that the algorithm will not find a new solution to what has already been assigned. So at that point, you will see that the iteration is finished because it cannot find a new solution for the assignments. Okay. Now, when your assignments are completed and the new solution cannot be found for this particular iteration, what the algorithm does is algorithm calculates a summed up variation of the assignment basically here. So the total variation within the clusters that is calculated, um, and that will give you an idea about, okay, so this is the total variation within this particular result, but the algorithm is not done yet. It needs to do all of the process again by initializing with some random points to be selected as the cluster representatives. Okay, so it does the same again, process the same basically steps again, and then this time you will see that the new iteration that the algorithm has performed, uh, the variation, the total variation of this particular iteration, it is lesser than the earlier iteration. So that means that there is possible that the algorithm is finding better solutions in a subsequent step. So the algorithm will not stop, of course, it will go on, and the iteration will go on, iterations will go on. And at some point, what will happen is that the algorithm cannot find a new better or reduced total variation from the earlier step. So at that point, your basically algorithm will be converged, kind of, right? So that was the process of k-means clustering, and this final result you can visualize how your kind of clustering has gone. Different tools, for example, if you have 1D, 2D, this kind of data sets, you can easily use for calculations as well as for the visualization of the cluster points. Um, you can utilize Python and its different tools that they have, right? The different libraries, they include the tools for k-means clustering also. And how you find out the number of clusters that is needed. So you can use something called the elbow method. So elbow method utilizes the within cluster sum of square values. And at certain point, you will see that even if you increase the number of cluster or the cluster uh, size, basically the number of clusters that you want to include, even if you increase that, your within cluster sum of square value is not reduced further, or it's not significantly reduced further. So you can take that as your representative number of clusters that you are using in the calculation. uh, this is basically kind of representation of what will happen if you do the WCSS as 0. So technically the goal of a k-means clustering algorithm is to minimize the WCSS value. So why not take the WCSS equals to 0, which means that for each of the data point, you are assigning those to be single representative clusters. Basically, each individual data point is a cluster itself. But as you see, it does not give you any information, because by considering all of the data points as individual cluster representatives, you cannot get any information about the groupings or about kind of what is happening, how your data is distributed, these kinds of information, right? Similarly, on the other hand, if you have only one cluster, your WCSS is max. So again, that doesn't tell you anything either. So it has to be somewhere in the middle, and that middle point is the elbow point, basically in this case. Okay. Um, the same sort of information or same sort of calculation is done by this class also. Again, another clustering method. So just remember these inputs, basically the inputs, and then how you assign the parameter, how you indicate the different parameters. Okay, for blast class. And yeah, so I mean, different structural information about alpha helices, beta sheets we went through, and then we saw that based on the composition of the sequences, so if you want to find out how distinct each sequence is or how different kind of multiple set of sequences in a pairwise way are, so you can calculate something called the Hamming distance as well as the Euclidean distances. So the composition based way, the Hamming distance is basically goes like this. So you just write two sequences, you lay out all of the individual amino acids kind of in the form of a column, and then you do a composition calculation, for example. So in this set of sequence, you will see that alanine is present 3 times. So that means that out of 10, you have 3 alanines. So that means the composition will be, or this fraction kind of, the composition fraction will be 0.3. So you do this for all of the possible amino acids. And then for the bottom case, the bottom sequence, you do the same calculations. And now what you do for the distances, you do a compositional difference. So of course, where you do not have any amino acid present for the sequence 1 and 2, you do not need to do those amino acid calculations, but for the rest of them, you just do a mod of comp 1 for the particular amino acid minus comp 2 for the particular amino acid. You take an absolute value, and whatever possible, you just do the calculations, you sum all of them up, and that will basically give you a Hamming distance in total. Okay, so remember this formula for calculating Hamming distance. Um, Sequence identity is kind of similar in this way. Just remember that when you have the identical residues, you divide that with the length of the shorter sequence. So for example, here it didn't matter because both of the sequences had the same number of residues, but let's say one had 8, another had 10, and if you find, for example, 4 identical residues within them, so then 4 by 8 should be the case, basically. So basically 50% sequence identity you can consider, because the lower of 8 and 10 is of course 8, right? So you divide that by the length of the shorter sequence. Remember this one in particular. Hamming distance again, and then another was Euclidean distance. So Euclidean distance is kind of similar approach, but it's a little bit more complicated. You do calculate the composition for the individual amino acid components wherever possible. But instead of just simply comp 1 minus comp 2, comp 1 of this particular amino acid minus comp 2 and adding them together, you do the comp 1 minus comp 2, you do a square of that, and then the output you do a basically summation, and you do a square ultimately. So firstly square, then the summation, and then the entire thing squared. So that will give you a Euclidean distance. Okay. So in this case 0.2, that is the Euclidean distance. So remember this formula also, Euclidean distance formula. Like I said, just go through the earlier slides. Wherever there is a formula uh, that is discussed by Professor Bhumiha, try to remember that basically. Okay. So just we again talked about non-redundant sequences. So basically, whenever you have uh, sequences without similarity, much similarity, then you can consider the data set as non-redundant set of kind of sequence, right? So yeah, so that was one calculation, and then another set of calculation you can do is you can do propensity calculation from amino acid sequences, let's say. So what is the propensity for a particular type of amino acids to be present in this type of secondary structure only? So in that case, you need to calculate two kind of fractions. So one is this type of residue, how many are present in helix. Let's say we are doing the propensity for a helical structure. So how many of this type of residue is in helix, and then how many type of this residue is in the whole data set. So you calculate a fraction of that in alpha of I divided by capital N of I, and then you do for all of the residues the same thing. So how many of all of the residues are present in helix divided by how many of all of the residues is in the data set, basically the total number of residues. So you do this n alpha divided by n, and you divide the first fraction by the second fraction, and that will give you a propensity. So here not only the type of residue information is included, but also the whole residue, the whole basically all of the residue structural assignment is also included here, and that will give you the propensity basically. Okay. Again, another kind of thing that you want to calculate from the amino acid sequences. So that was the GOR method, which is the Garnier-Osguthorpe-Robson method for information theory. Um, information theory is again it is kind of explaining the similar sort of thing. Basically, the information content of alanine in helix. Okay, so kind of similar to how many alanines, what is the propensity of alanine to be present in helix? Same kind of way. So you do calculate in a similar way. Remember this type of formula. So here again, you have to do calculate a couple of fractions. So one is how many of this particular residue is present at the secondary structure. You divide that by how many of this particular basically the total number of residues in this case, I guess. So for example, here it is saying for alanine among 100 residues, 80 are present in helix. Okay, so what you do calculate you calculate firstly how many alanine present in helix, so that fraction, and then how many of the alanine is not present in helix. So this curly symbol this kind of indicates not, basically a negative statement. So how many of them are not present in helix? So you calculate 80 by 100 and then 20 by 100 to do this division. So now you have one set of value here for ln calculation. For the other ones, again you do the same thing, but for all of the count of total residues. So how many of the total residues they are present in helix? So 600 divided by 1000, you calculate this fraction, and then how many of the total residues are not present in helix? That is 400. So you do 400 divided by 1000. You calculate this fraction, just do a subtraction, and then you can get the answer for GOR theory. Okay, so remember the formula for GOR as well. Information content basically. Okay. And yeah, so now I guess we moved on to different structure-based analysis. So these from until now they were basically the example of sequence-based parameters and sequence-based analysis. And now we kind of switch to the structure-based part. Okay, so we saw that for any structure that you have, so any structure that can be uh, let's say obtained from a molecular dynamics trajectory or any similar protein structure that you have available on the PDB database. So how are these structures different? How you can know that? You can calculate a statistical parameter called root mean square deviation. So this will tell you the difference, how these two structures are different from one another. Okay. Um, so that's one important formula or one important parameter. There are a couple of other parameters that will let you assess any structural changes. For example, radius of gyration as well as solvent accessible surface area, quite frequently used in a lot of analysis. But to delve into the structural part, we discussed what is going on with the PDB database basically. So that's of course a database where a lot of research groups when they calculate or when they basically experiment with the X-ray crystallography, and they get the coordinates of a certain biomacromolecule or protein ligand complexes. It can also be. So they submit those in the PDB database. It also has the information from cryo-electron microscopy also, which can also give you the coordinates, right? And we saw that how you can assess the quality of a structure. So you can assess them in terms of something called resolution. So it's kind of the same thing as your optical resolution. So whenever you have a lower value, that means the quality of the structure, at least from the resolution perspective, is greater. So in this case, for example, the option C, structure C, which is 1.14 angstrom resolution, it's the lowest of them all, the lowest value of them all. So that means it's the best resolution available for this particular structure. Okay, so it's opposite, the value lower, the resolution better. Okay, so it's a metric that reflects the quality of the collected data of a crystal for protein or nucleic acid. Um, Here it says that in an aligned crystal where you have identical orientation for the proteins, the high resolution data is fine and it can quickly, easily capture the diffraction pattern. However, if there is slight variations due to local flexibility, then the structure will lack the finer information. Okay. Uh, Typically, if you have structures around 1 angstrom, very close to 1 angstrom, let's say, those will be considered as very high resolution, very good structure. Whereas if you have a structure of 3 resolution, or let's say related to a little bit closer to those, for example, it could be 2.8, 2.9, or even greater than 3 angstrom. So those are usually not the best quality structures. They did not have the information or the coordinates for the all of the atoms that are present in the protein captured the best way, basically, or captured the best. So in that case, you will see that oftentimes those structures will deviate. If you try to do some calculations like simulations, those structures will deviate more because there was already some sort of a higher range of uncertainty present within these structures, initial structures, right? Of course, they will deviate more from this particular coordinates that you are getting from the structures. Okay. Usually, it's better that for a single protein, if you find a structure, you just do not go for this particular structure with your analysis directly or your calculations directly. You try to find out if there is anything else available also in the same database. If you eventually find a better structure, you can opt for that basically. So try to just go through the database, try to do different searches, queries, etc. Um, and then whatever outputs you are giving, these kind of PDB IDs for this particular protein, try to compare them and try to see if you get a good resolution one among those, right? So here is what was talked about by a higher resolution, higher kind of value resolution structure lacking the finer details. Okay, so this is kind of the electron density map for the structures, whereas you see for the 1.0 angstrom resolution, the electron density map is quite subtle. It captures the information quite well, whereas for the 3.0 angstrom, it does not. Right? And yeah, so that was on the structural part. Now on the structural part, um, we oftentimes saw that what you can do is you can, based on different parameters, you can calculate a free energy change. You can calculate free energy distribution also. So in this case, again, you have been given a question. So of 25 values, 5 of them are in surface. So that means the other 20 are not in a surface exposed region. So they are kind of in a buried environment, right? Now you are asking the transfer free energy of value. So transfer free energy in this case will be, let's say, how much energy is needed to transfer something from one set of environment to another. Okay. Now, how you do it? You do basically, as Professor Kumiha had given the example, you include this formula particularly for transfer free energy, where minus RT ln f of I is given, and then the f of I will be um, the number of this particular amino acid in a interior or in a buried position divided by the number of this particular amino acid in an exterior or in a surface exposed position. So in this case, you see that ln 20 by 5 was there, so ln 4 was there, and it seems that this energy you are getting that's minus 0.826 kcal per mole. Okay. Now from this particular statement only, you can see that if 5 are on the surface and then 20 of them are in a buried position. So that means, of course, valine is preferring a buried sort of environment, right? Uh, so in this particular case, what this fraction is indicating, it's indicating the transfer from an kind of surface exposed environment to a buried environment. Okay. So because valine is preferring a buried environment, you will see that the transfer free energy is coming in a negative uh, value. Okay, so minus 0.826, that means from exterior position or in a surface exposed to an interior position, the transfer is favorable for valine, right? So that's the gist of it. Just to add something to it, so we saw that these kinds of formula for free energy calculation where you do minus RT ln of something, some fraction. So that is also utilized in a free energy landscape uh, formulation basically, where you, if you have a certain data set, whether it would be 1D or a 2D data set, you can basically divide that data set into a set number of intervals, and then these intervals you can calculate how many population of data you have in a particular range of interval. So these intervals are kind of called beans, and then relative to the most populated bean, you can calculate the energy for all of the others. That will give you an energy surface. This is a pretty good way to also look at the energy surface of a protein and how it is kind of changing throughout your calculation for a simulation. Um, Basically, you will be able to visualize where a minima positions are and how much difference that minima position has with the surrounding, let's say, metastable states or relatively higher energy states that the conformation is passing through. Okay, so this sort of surface visualization you can uh, utilize a free energy landscape formula for that based on any kind of reaction point. Okay. Um, so that was free energy landscape and then energetics part kind of. And then another set of interesting calculations that can be done from any structure, those are contacts. So contacts, what they are, contacts are basically distances. You can think of them as that. In the 3D space, in the 3D structure, if you have the distance between two residues, or let's say certain atoms of these two residues, let's say C alpha atoms, right? If you have them in certain cutoff level, within certain cutoff level, you can say that these two residues are forming a contact, and it can give you idea about the communication path from one part of the protein to another. Let's say certain contacts are important to keep structural integrity at certain parts. So you can also find out this way that which distances are relevant in a lot of cases, right? uh, they could have some sort of function in regulating the overall conformation of the protein. So you can find out that as well. You can find out the communication pathway in a lot of sense. Um, and the contact map will be basically if you try to map all of the distances for these proteins together. You can just combine. You can try to combine all of the different contacts formed in a one kind of plot or graph or one data set, right? Uh, and let's say in a contact map or in a contact calculation, you can have broadly two types of contacts. One is a short range contact, and another is a long range contact. So a short range contact will mean that two residues that are present very nearby in the sequence, they are forming contacts. And then the long range contact will be two residues which are very distant in a sequence. For example, at position 4 and at position 40, let's say. They are very distant in a sequence, but the protein folds in a way that these two residues come closer together and then they form a contact. Okay. So those are long range contacts. And as we saw earlier also, that depending on the formation of short range contact and long range contacts, you can find out certain things about the structures of a protein. So in this question, of course, arginine 6, isoleucine 7, they are forming, they are of course just amino acids apart, one amino acid apart in the sequence. If they are forming a contact in the 3D space, then they must be forming a short range contact, right? So just remember this short range and long range contact uh, has correlations with uh, the different calculations, for example, short range order and long range order, which is also further correlated with your structural prominence of certain types, for example, alpha helical beta sheets, as well as the protein folding rate that we saw, right? So yeah, so you can calculate the percentage accessibility also, and yeah, this is one part that we went over. So basically, based on the presence of or dominance of alpha helices and beta strands, which are secondary structures, of course, you can divide the protein into different classes. So it could be either all alpha class, all beta class, alpha plus beta, or alpha by beta classes. So what are all alpha class? Self-explanatory. These are primarily formed of alpha helices. All beta proteins will primarily have beta strands. They will form the beta sheets. Um, and then based on how the alpha helices and beta sheets, if they are kind of equivalently present in this protein, based on how they are arranged, you can divide the protein into, you can categorize them as either alpha by beta or alpha plus beta. So what are alpha by beta? Basically here in this case, um, you have alternating patterns of alpha helices and beta strands. For example, beta alpha, beta alpha, beta alpha. Sometimes you could have a couple of alpha helices or beta strands together, but broadly you have them kind of in a separate way, right? They do not clump together. Same type of secondary structures are not clumping together. So beta alpha, beta alpha, beta alpha, these sort of ways. So those are alpha by beta. Whereas in the alpha plus beta, you will see segregation. So what segregation means is basically you have alternating patterns still available, but instead of single unit versus single unit alternating for alpha and beta, uh, mostly you will find that a bunch of beta strands, they are kind of clumping together forming beta sheets, and then a bunch of alpha helices are also present together, and these units as a whole, they are forming alternate patterns. So for example, in this case, the best example will be this one. So beta 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 beta, so 4 beta strands together followed by a alpha helix followed by again 3 beta strands together, and then basically the pattern kind of goes on in this way. Okay. um, yeah, so that's what was for the alpha by beta, alpha plus beta, and then these sort of classifications. And um, now we went over to something called homology modeling. So homology modeling is again one important part, uh, whereby you basically try to find a homologous set of protein, basically a homologous set of proteins which are homologous to, of course, uh, a novel protein, let's say, or a new protein that you have found out. You have the sequence for this one. You do not have the structure for this one. Okay. So now, based on what structure the other homologous proteins they have, based on that, you can derive the structure of this newly found protein, basically. So that is called something that is called as homology modeling. So firstly, you have a target sequence. You do a database search. You identify the best template for alignment, right? Then you do the sequence alignment. From the sequence alignment, you will get information where you can get the model framework, uh, just getting the backbone information from the aligned portions, and then you can add the side chains and loops. You can optimize the model and evaluate the model. In total, that is homology modeling. Firstly, you do the template selection, which is quite important, as it is kind of forming the foundation of your homology modeling. You do the sequence alignment, of course. Then what you do is you do backbone modeling, of course. So backbone modeling are basically your aligned parts. You will take the backbone kind of coordinates directly from what you are aligning with. Okay, so the information for the uh, the the template structure, that is already there. So the aligned part, you take the backbone information from there only, and then after that, depending on whether your particular, let's say, amino acid is matching between the target and the template, uh, basically between the template and the protein that you are trying to align, you are trying to find the structure for. If they are matching exactly, you can also copy the side chain information, coordinate information for those also. Okay. Now, for the loop models, if there is, let's say, gap present at certain part, you can do this via ab initio calculations. You can sample the different torsion angles that are generated. You can then basically, you know, discard all of the unfavorable steric positions, and then you can get a model for the loop in that way. You do the side chain refinements following that, and these are done by different protamol libraries. Uh, and then you do model refinement if needed, followed by evaluation. So on evaluation, you have certain tools such as ProCheck, which you can use to check if your um, model structure is basically good enough for further calculations or not. Okay. Now, since we were on the structural part, we discussed again very uh, related advancement which came from the CASP basically competition, right? So CASP is of course critical assessment of protein structure prediction. We briefly talked about its history, and we showed that basically in CAS 14, which is in 2020, um, AlphaFold 2 was entered in this competition, and at this particular point, the threshold that was set by the organizers of the CAS. So basically, the threshold that they set is that you have to predict a novel structure with a 95, a 90 score, basically a score better or at least 90. Now what they do is basically they have already previously uh, they previously keep information for a certain protein that is new, for which no data is published, structure related data online, not publicly available basically, but they themselves have sort of uh, done or they themselves have solved the actual structure in the crystallographic way, right? They have that information, they provide use a sequence and then say that okay, now use your algorithm and predict me a structure, and based on how it is closely matching with the experimental data, experimental structure, you will get a score. Okay, your model performance will get a score, and the score of 90 was particularly exceeded in CASP 14 2020 for AlphaFold 2, which was kind of a revolutionary uh, basically advancement in the field of protein structure prediction, right? And then again, we briefly discussed that, of course, the Google DeepMind, which devised AlphaFold 2, the lead AlphaFold 2 researcher, John Jumper, and then CEO of DeepMind, who kind of oversaw the entire project, the name is Hassabis. They both were awarded one half of the Nobel Prize 2024 for chemistry, right? And then the other half went to David Baker, of course, for computational protein design. So computational protein design is basically you try to design first of all the sequence of a protein and then the structure of the protein also, which does not exist in nature. So you can design according to a particular function that you want it to perform. So um, that is the motto there. Okay, so as he has said sometimes in his speeches, that if you basically let evolution run through, if you let evolution run its course for a lot of problems, you will eventually find out some solutions. So biologically, we'll probably generate some sort of biomacromolecules like proteins that will take care of a lot of issues that we are facing right now. But evolution takes longer, much longer, right? Thousands and then lakhs and millions of years. So we cannot wait that long. So we have to tailor certain designs or certain novel ideas to the functions that we want it to achieve now, basically. So in that sense, you can, for example, design proteins that can, let's say, degrade plastics. That's an environmental approach. There is designing for novel proteins related to neutralization of, let's say, snake venoms. So that is one approach. So different cases, you can get different kind of approaches. So that is the part of computational protein design. um, and if you want to look further, to look into the videos that I have sometimes linked in the slides, and then go through some of their papers and the tools that they have developed, particularly Rosetta Fold diffusion, etc. So try to see what those tools do, of course, alongside AlphaFold 2. So you can see basically how the process or how the workflow exactly works. Okay, just some examples of the papers from both of these labs. Okay, is given here. Yeah. And also we saw based on fluorescence intensity, we can do urea denaturation experiment, which can tell you the fraction of basically the free energy change as well as the different fractions of folded versus unfolded proteins. Again, very similar formula. Just try to remember these formulas and the free energy change formulas. We quickly discussed about different interactions that are used for modeling of the structures for proteins and the different biomacromolecules. One is van der Waals forming Lennard-Jones potential information, which you do not need to remember, but it kind of looks like this. You have attractive as well as a repulsive potential information embedded in here, and this van der Waals potential is always used in any sort of simulation force field inside that. Okay, again something regarding urea denaturation. So free energy change, again always remember these sort of formulas are there, minus RT ln K, but which fraction you put in K, that matters here basically, right? Yeah, and then we discuss some more on the structural sort of parts. So basically, uh, one of the calculations that always comes in the setups, that is distance calculation. And how do you do distance calculation? You have the coordinate information for all of the atoms in a system, right? In a PDB structure, in a MD snapshot output, etc. Um, and you have the basically the XYZ coordinate information. So you have where this particular point or where the atom is in space. You have that information. Uh, Now, if you have two atoms differently, right? For example, one has x1, y1, z1 as the 3D coordinates, another has x2, y2, z2 as the 3D coordinates. You can calculate their distances using this formula. So it's root over of x2 minus x1 square plus y2 minus y1 square plus z2 minus z1 square. Um, and then based on the distances that you are getting between certain atom pairs, you can find out whether these are forming certain types of bonds or not. For example, we know that cysteines, they form disulfide bonds. Uh, so basically, you can calculate distances between the different sulfur atoms, and you can find out, okay, these two cysteines should form a disulfide bond in the system, right? So again, the same thing here. You see that 23 and 97 cysteines, they had a distance of 2.046. The other one, 138, 192, 2.051. So they are in disulfide bond forming range. Okay. These ranges can be a little bit uh, different across different tools. For example, one can state that around 2 angstrom, one can state 2.3 angstrom, 2.5 angstrom, so on. Just remember that this can be within ranges that differ in basically different tools. Okay. Uh, so disulfide bond is one sort of formula or one sort of distance, and then remember there is some also called charge-charge interactions. So if the two charged residues, they are coming very close at around 4 angstrom or uh, lower distance, so they can form basically salt bridge interaction. That's another one that you can try to remember. Um, and then on the assessment of structure, of course, we talked about the moment of inertia and the related radius of gyration. Radius of gyration particularly tells you if a structure is getting or getting more or less compact in a sense. Okay. Uh, yeah. And then Professor Tumi had discussed about something called average assignment. So the average assignment is very simple. It's that you want to change one amino acid to another, uh, but you have already available data of similar sort of amino acid changes, for example, valine to alanine, and what happens, what is the free energy change among those. Okay. So if you have that information, what you can do if you want to assess a new in a different position valine to alanine mutation, you can do a average of all of the available free energy values, and that will get you an idea. But average assignment is of course not that great in exactly determining this, because we know that uh, the free energy change, they can be favorable or disfavorable based on where the position of these amino acids are. Okay. For example, a valine to alanine mutation will be favorable, um, can be favorable somewhere where there is, let's say, more tightly kind of packed structure. It can be, again, if the side chains are increasing in size following the mutation, and the structure is already quite tightly packed, then a mutation of that kind will also induce a lot of steric hindrance. So in that case, um, basically steric hindrance based disfavorability you will see. So it depends on what type of amino acid is being mutated to what type, and it depends on what is the surrounding environment basically. Yeah, one other structural parameter that we went over, that is the B factors. So B factors indicate flexibility. We saw one column is always there in the PDB files. If you open it up using some text editors, that gives you the B factor values. There is also normalized B factors, whereas you calculate the B factors for the certain residues basically. Okay, and this is basically B minus B mean divided by the sigma, which is difference deviation. And then if the normalized B factor is greater than 1, it is flexible. If it's less than 1, then it's a digit um, residue basically. Okay, so greater than 1 flexible, less than 1 is a digit residue. and then some sort of kind of descriptive and analytic type of question. So we know that predominantly the folding and the stability of such folding for proteins, they are primarily indicated by hydrophobic residue. Of course, at certain parts you have certain charge interactions which keep in place, let's say, certain secondary structural part or certain folding via a long range order contact links, a long range contact, right? So those can keep certain parts in place, but mainly the hydrophobic residues or the hydrophobic clustering is what drives the folding of a protein. So that is what I was asking in this case, um, which is highly correlated with stability in buried mutations. Of course, that is hydrophobicity, because in a buried environment, uh, you do not have access to water. So that means that the higher hydrophobic a residue is, the higher will be its propensity to get into a position where a buried surrounding is present, basically, right? So that's the correlation that was mentioned here. And then as I just said, it matters which type of mutation is occurring, and it matters uh, what is the surrounding environment, basically. Okay. So we see that a alanine and a valine residues case. So valine has a uh, large volume secondary, not secondary, the large volume side chain, right? So a larger volume side chain, that will mean that alanine to valine mutation in a already tightly packed environment, that will be disfavorable. Okay, because again, if it is changing into a higher volume, so that means it will increase the steric hindrance or steric clashes at this particular position. So what can also happen is sometimes the mutations can be space filling. So space filling will mean that there was already space for the amino acid to, let's say, somehow mutate and increase in size. So it had a quite loose packing, and whenever you change it to a higher volume amino acid, higher side chain volume, so it will fill up this space. Filling up the space will mean that the contacts will be improved in this position, and it will go more towards a tight packing and usually a more stable structure. So this is a stabilizing mutation, a space filling mutation. Destabilizing is, of course, when you have collision, basically, or static hindrance. Uh, and then if your certain residues were taking part in charge-charge interactions, so if you suddenly change the charge, or if you mutate one amino acid to an opposite charged amino acid, so it can, of course, easily destabilize the region or the entire protein. So those are depending on the surrounding environments, of course, right? And what particular role this amino acid is playing already. For example, in the case of charge-charge interactions, right? And just based on the information and based on, let's say, free energy change from one to another, uh, depending on what is favorable, what is unfavorable, you can easily find out what the previous position was and how the current kind of status of the situation is at, basically. So in this case, for example, positive mutation and the negative mutations were indicating stabilizing and destabilizing. So don't go for the symbols just for the sake of the question. It's positive stabilizing, negative destabilizing. Since it's alanine to valine mutation, so that means it's a side chain increasing mutation, right? So for side chain increasing mutation, you saw that wherever you have the basically the most negative value earlier, which was for the alanine 42 to valine case, so the 42nd position, basically. Um, the most negative value in this case, that would have indicated the most tightly packed position, because since alanine to valine is a volume increasing mutation, so that means a negative value, as in this case destabilizing scenario. So in this case, that will indicate the most destabilizing scenario will be the most tightly packed position, because that will cause more steric hindrance, right? So these type of analytical questions you can also encounter, whereby you have to correlate certain free energy changes. You can correlate the change in the amino acid that is going on. Based on that, you can find out what is particular going on at a certain position, basically. What is the packing or these kind of parameters for a certain position in the protein. Okay. Binding propensity calculation, you can also do kind of similar numeric questions. Um, and then the chevron plot we also briefly discussed. This is kind of the folding kinetics plot. So kinetics of protein folding, remember, that is done via the chevron plot. Uh, and then for all of this, yeah. So the contact order also. So we saw that the contact formation can be based on the 3D distance of the 3D space between different residues. So you can take the distance, for example, from C alpha atoms. Um, and then you can calculate the contact order and the long range order from those. So contact order formula is, for example, this one. Now, higher contact order means, of course, higher long range interactions are being formed. Uh, and in that case, you see that the protein folding rate for the long range, mostly long range interaction dominant proteins. So you'll see that this folds slower basically. The protein folding rate is slow. So if the contact order is high, folding rate is slow. If the contact order is less, folding is fast. So they have a inverse sort of relation in this case. The same thing is observed in the case of long range order. So earlier one was contact order. This is long range order. Slightly different formula, but kind of the same concept here. That is also negatively correlated with the protein folding rate. And yeah, basically just remember this one, that they are negatively correlated. Um, and then we just quickly switch towards the other important factor or part of the computational kind of analysis, which is the drug discovery, right? So I guess Lipinski's rule was mentioned there. So it's a part of ADMET analysis. Uh, Particularly, you can use this as a filter to discard a lot of uh, candidates from your big list, basically big list of candidates. Okay, so you can filter using the Lipinski's rule, which states that the molecular weight should be less than equals to 500, the log p, log of octanol water partition coefficient, that should be less than equals to 5, the number of hydrogen bond acceptors should be less than equals to 10, number of hydrogen bond donors, that should be less than equals to 5. And if there are basically less number of violations, for example, it's one violation or maximum of two violations, it's a less than two. So it counts one violation as still good. So you can proceed forward with that for your further studies, basically. But just the disclaimer, which is that Lipinski's rule was devised a long time ago. So a lot of FDA-approved drugs that we have seen over the years, they violate Lipinski's rule in a lot of cases. Okay, so it's not the end of all. You will see that different pharma companies, they have devised their own parameters. For example, the Pfizer rule, the GSK rule, etc. So you can look into those as well. And usually some server where you have extensive kind of ADMET calculation tool set, like ADMET Labs, Swiss ADMET, ADMET SAR. So they will usually indicate multiple filters or the result for multiple sort of uh, this sort of basically a standard parameter calculation or standard rules. Basically, there's also multiple of those, and then the individual components also, so that you can decide for yourself which molecules you want to go forward with. Yeah. And then of course the major part where the admit calculation is coming in is drug discovery. So in drug discovery we see that, of course, molecular docking we discussed, um, and the three terms were included there. One is binding site, which is the region where the molecule is binding. One is binding mode, which is the particular geometry of the ligand at the binding site, and another is the binding free energy, oftentimes indicated in kcal per mole or kilojoule per mole, which is basically the free energy of complexation. So initial minus final, final minus initial. Initial is receptor and ligand separate, so the total energy of them. Final is when they are in a bound form, so that's the complexes energy. If you do the final minus initial, you will get something called the delta G, or the binding free energy, uh, which will give you an idea about how favorable your binding is, basically. Okay, that's the example there. Uh, Again, same thing. You can do the calculation of the binding free energy in a variety of ways, of course. The docking also give a type of score which is reflecting binding free energy. Otherwise, from a extensive calculation from simulation, you can do free energy perturbation, free energy thermodynamic integration, um, and also less extensive ways such as MMPPSA, MMGPSA, which I have mentioned sometimes. And basically, you get to the value of enthalpy, which is composed of different smaller kind of individual components. But you have the value of enthalpy, and then if you have the entropy contribution, you can calculate the binding free energy from those. Okay, using the Gibbs free energy calculation formula basically. Uh, and then that leads us directly to what is called docking. Okay, so molecular docking is basically you have a molecule, small molecule, let's say. You can do protein docking also, but let's say we just discuss using small molecules. You have a small molecule, you have a target binding site where a cavity is present. You want to fit this small molecule into the target site. You generate different poses for that, and you try to see, okay, which pose is scoring better. You want to find out if there is one pose which is give you a good enough docking score inside this binding pocket, so that you can say, okay, this molecule is binding to this particular target protein. Now, why is that important? Because in physiological sense, of course, the proteins use their catalytic sites or any kind of functional site for signaling purposes, right? Cellular signaling. So if you block the sites, the signaling is cut off. And then if the signaling is the cause of some symptoms or some disease, you are basically preventing that. Okay. So that's the reason why docking is important. Uh, on a broader scale, on a kind of broader theme, it's significant because uh, rather than going for some random molecule and testing it on wet lab, you are previously finding out from a large set of structures using some sort of screening, high throughput screening. You are basically initially trying to find out whether the molecules that you will get, the hits that you will get, will actually bind to a protein or not. Okay. Similarly, if you have a couple of molecules also for going for experimental processes, you can try to find out using docking whether this molecule is binding to that protein or not. Okay, so it kind of cuts down on the exhaustiveness of search space, you can say, uh, basically on the size of the search space, whether which hit will be better for your further studies. So that kind of thing. Okay. Now the two elements of docking are basically the search algorithm and the scoring function. So the search algorithm is what generates the different poses or the different geometries at the binding site for the ligand, and the scoring function is what scores those different conformations, so those different geometries. Okay, you can do rigid versus flexible docking. Rigid docking, you keep the ligands as rigid, you keep probably the proteins as rigid also. In the flexible docking, you try to do insert flexibility in all of these cases. Okay, you can also in some cases see which bonds you are trying to keep rigid, which bonds you are trying to uh, get flexible, which is basically allowing flexibility, which amino acid residues you are allowing as flexible, so on and so forth in different tools. We briefly went over, Professor Rumiha had gone over basically the Autodock tool for docking. We briefly talked about the implementation of Autodock FINA within UCSF Chimera, which is a very simple way to do the docking. But I guess you should use Autodock FINA or Autodock directly because that will give you a more extensive set of tools for your calculations. Okay. Uh, yeah, the different algorithms, basically stochastic search, hybrid global local search algorithms, and then the related factor or the related type of calculation was QSAR, which is quantitative structure activity relationship. So this is based on a predefined or a kind of already available data of structure versus activity. So basically, you have for certain, let's say against certain protein, you have a set of molecules that are already showing good activity. You have something like some binding affinity scores of them, and then what you do is that you convert the molecule to some sort of structural representation. Let's say you extract some features or descriptors from these molecules, from the structure of the molecules, and then you train the model using that, and then finally you can use that to predict um, the new ones, basically, right? That's the part of QSR. Um, for docking, there could be different scoring functions, force field based, empirical, knowledge based potentials. By the way, remember these examples. Okay, so these are the scoring functions particularly used in certain tools. I think Professor Kumia explicitly gave these examples, so you can remember this. Yeah, so empirical scoring, force field based scoring. So force field is quite important because it's basically embedding the potentials for the different interactions that can occur within this force field, and then you can use that to calculate the new positions in the case of simulations also. So quite important in simulations as well, apart from docking. Yeah, we briefly talked about genetic algorithm. Genetic algorithm is basically a way to find out your conformation or ligand geometry within the binding site, right? It's a stochastic algorithm because it includes the randomness in the particular calculation also. So basically, you find out or generate random poses, then you score them, you find out the better scoring ones, you call them as parents, the parent poses. You take certain parts of those, combine them together to create new poses, the new generation of poses, and then you keep doing that basically until and unless there is no improvement in the scoring. So at that point, your algorithm will be converged. That's one way to find out your uh, basically the search space or the particular ligand conformation for the docking basically, right? And then briefly we talked about computer drug design, how it works. So the first step is, let's say, the screening part. If you have a large set of data, you can use a high throughput docking process for the screening. You can weed out all of the unnecessary or the unwanted molecules which show lower binding affinity. You can take the hits, you can go for admit calculation. You can already use admit based filter beforehand, but you can go for a thorough admit calculation, admit analysis, basically prediction. And then based on that, whichever hits you get, you can go forward with that with simulation and free energy calculations. Again, a little bit more rigid or rigorous set of approaches. And then again, if there also you see good results, then only you proceed for experimental validation and whatever is coming further. Okay, that's the part of the computer-aided drug design process. Uh, and we briefly talked that instead of doing normal docking nowadays, if you want to screen from a large means ultra large billion scale library, you can use deep learning methods such as the deep docking approach that is defined here in this paper. So basically, you only dock a subset of the molecules in a data set in the large library, basically like zinc, enamine, etc. And you use that docking score for the training of the model, and you use the rest of the database for prediction or for basically finding out which molecule is better, could be better. So that's how you do from an ultra large library using this thing called deep docking. And we talked about that nowadays artificial intelligence is not only in the part of the initial part of docking, for example, in target identification, target discovery, but also going forward, it will have more and more importance in all of the other aspects of it also. Preclinical studies, clinical trials, different biomarker discovery, evaluation, safety evaluation, even post-market monitoring, etc. In all of these cases, um, AI can come in handy basically. So the key takeaway is that if you have interest in, for example, further computational research or anything in this field, um, anything in the field related, or if you want to supplement your experimental research even, I would say try to learn AI and the new advancements that are being made worldwide from different research groups. Okay, so try to get through the different concepts, try to implement them in your research area, and that will be quite good for all of these cases basically. Yeah. And finally, I guess we kind of concluded based on the evaluation parameters for different models. So we talked about the different concepts, for example, true positive, true negative, false negative traits. So any kind of model, machine learning models also, they will require this sort of evaluation. Um, Again, remember these formulas, TPR, FPR, as well as sensitivity, specificity, and the accuracy calculations. Because yeah, these are latest lectures. So very crucially, you can get this kind of you can encounter these sort of concepts in your exam also, right? And also remember that one thing where you, instead of just explicitly stating out, you represent the true positive, true negative, false negative in a matrix kind of fashion, which is called the confusion matrix. So you can try to go through that as well. Okay. Yeah, so I believe that is what covers the entirety kind of the recap, I guess. Um, so yeah, that's the entire thing. We'll try to upload the videos as soon as possible. Um, And then for exam related queries, of course, you can always contact the NPTEL platform and the admins of the course through the forums. Yeah, I guess I'll just wrap up today. Again, thank you all for joining. Thank you all for joining all of the classes, lectures for both of the TAs, as well as for attending the lectures for Professor Komiha also. And then, yeah, all the best for the exams, upcoming exam. That is kind of the biggest part of your marks, right? So yeah, all the best for that. And thank you all. We'll see in some other course maybe. Yeah,
Thank you.
yeah,
Thank you so much, sir,
yeah. Thank you
for
so much.
this journey. Thank you so much, sir.
Yeah, yeah. And by the way, I in the Google Drive, I think I included some of the other resources. I believe you might have gone through it. So different books, uh, I guess relevant ones, and their PDFs, etc. Some papers maybe. So I had already included that in the shared Google Drive folder. So try to download all of those. I believe that those will come in handy in your studies or research or in any similar activities. Okay. So go through that, the relevant sections, for example, the ML-based books, etc., the coding kind of books, etc. So those could come in handy in a lot of cases. Okay. Yeah. So thank you all finally, and all the best for the exams. Thanks.